Well, why don't we go ahead and get started? So yeah, it's my pleasure to introduce Andreas Horn, who is with us from Brigham and Women's Hospital in Boston. He has an MD from Freiburg University and a PhD from Charité Berlin. And he's in the neurology department at Harvard Medical School and is focused on deep brain stimulation. And so he's been looking at the relationship between the connectome and stimulations and treatments and things like that across various disorders. So really important work, trying to see if we can actually leverage neuroimaging with an intervention to do something useful. So really interested to hear what you're going to be telling us today, Andreas. Thanks for joining, and I'll pass the baton over to you.
Thank you so much for the invitation. It's a big honor to speak here, and also thank you for the kind invitation, kind introduction, Vince. So it is true. We focus on deep brain stimulation as a maybe turning angle of the lab. I think the rest of it, I would say, is probably very much typical neuroimaging stuff. So connectomics and so on, and I'll talk about that. But there's always this anchor point of maybe a brain electrode in the brain and how it's connected to other sites in the brain. I do have I have started to consult for some of the DBS companies only if I can actually help maybe create better products, or have the feeling that this might lead to patient improvement sooner than later. And so I have these disclosures. None of them are directly related to the work I'm going to show, but these are companies that are in the deep brain stimulation field, and I sometimes work with R&D to help behind the scenes to improve therapy there. All right, so the broader topic I would say is neuromodulation, because I think a lot of what we are doing with invasive deep brain stimulation is also in a way applicable to non-invasive forms of neuromodulation. And I think one reason is that neuromodulation has some causality aspect to it. We do something to the brain, right? We interact with it, and we typically interact with a specific site in the brain. So at least when it comes to that site, we can probably say, okay, this was a causal thing that we introduced, and that leads to an effect that can be helpful. And so we can think of the broader concept of neuromodulation, as non-invasive forms, TMS, TDCS, and other forms, and then invasive forms. I think we can see deep brain stimulation here as one of the key technologies, but there's also lesional neuromodulation, where we actually carry out a small lesion into the brain. New rising star in this array of things is MR-guided focused ultrasound and HIFU type, so the high intensity focused ultrasound, where we actually burn small holes into specific parts of the brain with the idea that we tune down a specific circuit that has become overactive, dysfunctional, and so on. And it may surprise some of you that deep brain stimulation does something very similar. So we should think of DBS, at least from afar and as a rough explanation, as a lesional therapy, but with the key exception that we can turn off the lesion again, right? So essentially we're implying, we're adding a high frequency electrical field onto a small part of the brain, and that has multiple effects, but the key effect that leads to symptom improvements is very similar to this, making a lesion at that same point. So we think of it as a disrupting information flow in that part of the brain. So stimulation is really a misnomer. It's maybe one take home from this talk, that we should think of DBS as introducing small informational lesions that we can switch off again and test. And so this is just for people that have not seen DBS at all. This is a bit the idea. So electrodes are implanted into the brain and they stay there. So it's a permanent thing, often for multiple decades, 20 years or so. They can stay in here. And then there's a pulse generator, looks like a cardiac pacemaker in the chest, and it delivers through extension cables that go under the skin. So it's all in the body. They deliver weak electrical pulses, so patient don't feel any pain or something. It's not, something you can perceive. And they are typically 130 hertz, and they're typically very short pulses of 60 microseconds even. So short pulses, 130 times per second, and that leads to some sort of disruption of the information flow. And it's always fun to see the fantastic effect of DBS. This is of course a good responding patient, a top responding patient even, that I had the pleasure to treat in at Charité when I was still in Berlin. And the only real difference between the two videos is it's the same time point, but DBS was switched on versus off on the left and on the right. And we can see that the bradykinesia, so that the rapidity of movements, alternating movements, is much, much better on the right. And there's no dopaminergic medication involved. This is a patient with Parkinson's disease. And so, drastic effect. And it's really each time, even for people that have been in the field for a long time, miraculous to see that this happens more or less instantaneous with some symptoms, and we switch it on, and then the symptom goes away or gets much better. This is the operation scene. A small joke here, of course, but the idea is we use planning software from companies, FDA approved, CE marked, to plan exactly on the MRI. So first contact with the imaging system, and then there's typically a frame on the brain, and we can, the software gives us coordinates of where to put the electrode exactly. And that is not me, that's neurosurgeons. So I should mention that, and also not Angela Merkel. And it's usually, of course, in a sterile environment. And so the idea really is that, maybe from a surgical perspective, the procedure is not that complicated. It's a small burr hole, goes down, but really the magic kind of happens in the MRI and imaging and in the planning. So the surgeons really need to know where to put the electrodes, and that's not trivial. So they are often more of the neurological type of surgeons. They're really interested in function of the brain. It's a fantastic procedure. And so what I realized when going into this and trying to localize these electrodes across coordinates, across cohorts, is that we learned that millimeters really, really matter. Of course, everybody knew that the electrode needs to be in the right place to exert function, but it's really about small differences. So we have, I think, as a field, converged to this specific site here in the subthalamic nucleus as the optimal spot in general for Parkinson's. And this electrode, patient 21 in this cohort, was very close by to it. That's the active contact. So we have 4 contacts. The active one was this one, very close by. This one is just around 2 millimeters apart from the other one. But we see that the clinical improvement of symptoms is 80% for that 21, and then only 25 or 30% for patient 51, and then even worse if we're far off. And we've and others have shown this a lot of times. So it really seems to matter, and it's not just about roughly being the right spot, but you really want to be precisely in the right spot. What we can also see here is that these electrodes have multiple contacts. And with the pulse generator in the chest, we can later switch on, different ones after surgery, right? So over the 20 years or how long the patient has the device, we can still make adjustments. And we can also, switch on different combinations of contacts. So we can switch on multiple ones, have bipolar stimulations and so on. And then these are old electrodes, and the newer ones have 8 contacts or sometimes even 16 contacts now. So then it becomes much more complicated to do this in a trial and error basis, right? Here we could just switch on each one separately, test it, come to the conclusion that one is the best. But if you have 16 contacts, that already becomes more cumbersome also for the patient, because each time they have to, finger tap and so on. But it also, there are combinations as well. So that then, opens it up to a myriad of possibilities, and that's where maybe imaging and automated programming could come in. But as a first step, we learned from this slide that we as neuroimagers that are interested in this have now to be very meticulous as well. So as the surgeons, they really want to go to the right spot. We have to be very meticulous when reconstructing these electrodes, because millimeters matter. And often our images, as you know, T1 MRI is well resolved with 1 millimeter resolution. But if 2 millimeters already make a difference, that's then 2 pixels in the image space. And not only do we want to know where the electrode is in a given patient, which seems more straightforward, and even that is, by the way, challenging sometimes. We also want to make it comparable across patients, right? So we want to register them to a common space, and maybe different from the field of fMRI, or so millimeters really matter. So I think in an fMRI blob, nobody cares about 2 millimeters, but here we do. So that's what makes this quite challenging. And so a lot of this is really boring structural MRI, knowing anatomy, knowing where the CT that shows the electrode aligns with the MRI, and then how the MRIs of different people align with normative spaces. But I would say maybe in that realm, it might be a royal discipline because it's really so much about the details. And sometimes patients even have atrophy and so on. So it can be quite challenging. And to facilitate this process, we've created a software that's openly available called LeadDBS that I'm super happy about. Is now being quite used by many, but even more happy about that also others have joined in the development. So in case there's anybody in the audience that likes this concept and likes developing toolboxes and open source software, do reach out. We need help. It's a small community, and we would love to. There's so many things to maintain and features to develop, and so but that's the idea, right? So we want to know where the electrodes are, and then to get to the main point of the connectomic DBS, we want to then relate them to networks. And I think it's a natural assumption that most of you will probably agree with, that we need to know the brain circuits that are faulty first to then define the sites where we want to stimulate them. And with stimulate, again, remember, I mean maybe tune them down. So we have to define these brain circuits first. And once we understand those, we could know, okay, we now hit a specific node of them, maybe where they all converge or something like that. But defining these brain circuits is not trivial. And so the same applies in maybe a different way to TMS sites, so to other forms of brain stimulation as well. And big question is really how to define these circuits. We've used different things as a field. Of course, there's a lot of animal models, including optogenetics and so on, to understand things, but you never know the translational value between model organisms and humans. For example, in psychiatric disorders such as OCD or depression, it might not be that the depression models in mouse actually mean the same thing. And even in good models like the Parkinsonian models, there's still, the gap of anatomy between mouse brain and the human brain. And then, of course, we have used functional imaging, and we still do, and that's a fantastic option. We can scan Parkinsonian patients, we can scan healthy controls, we can contrast them, and we see a difference. But we never really know, is the difference going to be causally leading to the symptom, or is it even compensatory of the brain, or is it just spuriously correlated with the symptom emergence? So there's maybe a causality gap there. And then if we use electrophysiology, even there's typically correlative evidence. And I'm not saying that this means these three things are bad or unnecessary or whatever. They have very much have their value, but there's also maybe not a very direct translation into defining the circuits that we want to treat. So we came up with a different strategy in the center to reverse the problem and use the DBS sites and TMS sites in the first place to define the brain circuits, which will also not solve every problem, but is just a different way of trying to get to that answer. And we also, in some way, use brain lesions that naturally occur, for example, during stroke, and then lead to a specific symptom, or in rare cases, even get rid of a specific symptom to define brain circuits that might be more causally involved with the occurrence or the cessation of specific symptoms. And the general idea here is to essentially do what I just told you. We localize these electrodes as precisely as we can, and then we would often model around them the stimulation field, sometimes referred to as the volume of activated tissue or the electric field. And we do so using, advanced finite element method concepts where it's very similar to the EEG field with the forward model of the EEG field. But again, it's a bit more about the millimeters, so not about the bigger dipole structure in the brain, but rather about what happens around the electrode. And we then define sometimes axon models, sometimes more heuristical options to define a volume that's activated or modulated by DBS. And then we can use that as a seed for connectivity analysis. And as I think many of you know, there are these two main techniques that we could use. One is resting state fMRI for more functional connectivity types. There's diffusion MRI for estimating structural connectivity. And I'll focus a bit on diffusion MRI here. And I want to say that diffusion MRI is a fantastic method from a physical standpoint of view. It's a non-invasive method. We can look at brain tracts. It's fantastic. But it's also, anatomically, it has big problems. So sometimes we don't even rely on actual diffusion data, but we rely on atlases, such as this one. It looks like a diffusion tractography result, but it's not. It's actually a manually drawn atlas by anatomists that is informed by histology and their expert knowledge, where they had, holographic lenses on. And they essentially debated where exactly would these tracts go in 3D space. And so it's more to be seen as a three-dimensional anatomical atlas. But it's digitally represented, looks like fiber tracts, so we can use it in the same way as a diffusion result. But we often use also real diffusion data. And so we came up with a technique called fiber filtering, where we take all the tracts that are embedded in a connectome or in a pathway atlas, such as this one, and we go tract by tract. So we take each one out, similar to many techniques going voxel by voxel. Here we go, fiber tract by fiber tract. For each of them, we relate them to the field of the brain stimulation electrodes. And there are then several scenarios that could emerge. One would be that the top responding patients all modulated this tract very strongly, and the poor responders did not. So then, based on this, it's kind of obvious that, or, we can infer correlative form that this is a beneficial tract. And then the opposite would also happen, could also happen, where all the poor responders modulate the tract, but top responders did not. So we can maybe infer it's not an optimal tract. And then in some scenarios, it's really a mixed bag. The jury is out for this tract. It doesn't seem to matter. It doesn't seem to, preferentially be modulated by top responders or poor responders. So we say it seems like an irrelevant tract, and we filter it out. And so we can now do this at scale for every single fiber tract, and then magically we, get rid of some tracts that were irrelevant, and we retain the ones that correlated with symptom improvement and the ones that did not. And so the ones that did correlate positively, we typically, make red in our case. And in this case, this example, this would be tracts associated with tremor improvement on the right hemisphere of the brain. And we see that the cerebellothalamic tract comes in and gets selected, which is quite well known by now, and maybe one of the real findings that has been replicated a lot of times, that this really is kind of true. So now again, think of the idea that I said in the beginning, that we think that the brain stimulation acts more or less like a lesion. It's not exactly like a lesion, right? But for the time being, it's enough of an approximation that it kind of acts like a lesion. And these are just some examples of historical procedures that have been done, lesions to the same structure as DBS, and kind of led to the same effects, right? So it really holds true almost everywhere we do this, that it has a lesion-like effect. If we think of that now, then we could say, okay, what is this red tract now really? Because we essentially lesioned it for the symptoms to improve. So it's not a tract that helps to get rid of tremor if we stimulate it, but it might be the tract that actually led to tremor in the first place because it was somehow dysfunctional, and we, by lesioning it, got rid of the symptom, right? So to me, that's a very important thing to consider. I'm not saying we have absolute proof that that's the case, right? It could be more complicated, but it's not a tract that we stimulate or upregulate and then it helps tremor. It's the opposite. We downregulate it, and that seems to help tremor for whatever reason. So one inference from that could be that it was involved in maybe creating tremor in the first place, and was overactive, was dysfunctional, and by lesioning it, it helped the brain to get rid of tremor. So one conclusion, not a fact, but our current working hypothesis would be this was a dysfunctional circuit. And so I'll walk you through the first real example now, finally, which is Alzheimer's disease. So this story, I think, is very interesting. Should make the history books of neuroscience, and I'm not involved in that part. So it started before my time when Andres Lozano and Clement Hamani were in the surgery room. They were operating on a patient with morbid obesity. They switched on the electrode, added current to it, and the patient began to talk about a scene that certainly came as a flashback. Was seen outside, and he felt like this would be a scene he recognized from his distant past. And then he switched it off again, and the scene disappeared. Switched it on again with more, added more voltage, and the scene came back and then became more vivid. There were more details in it when they added more volts, and then even added more volts. And at some point, the patient recognized, oh, this was actually a scene that happened at night. So it seemed like a memory to the patient, and it was reversible. And that then led Dr. Lozano to think, that's the way he talks about it in his talks, where are we in the brain? We're close to the fornix, and the fornix is part of the PAPE circuit. So this could potentially be a treatment target for Alzheimer's disease or dementia. And so this actually led to a first trial with 6 patients that were operated back in the day from his team, and then a phase 1 trial as well, and a second phase trial that after that we came into the picture and were fortunate enough that we could collaborate with Dr. Lozano's team to look at the relationship between placement of these electrodes and the outcomes. And this is exactly the type of cohort that's very hard to register because Alzheimer's patients have atrophy, and we've tried a lot of things, but we came to the conclusion that we actually have to manually refine the warp field using a software we have created that's called warp field, where we refine the brains in such a way that they are better registered in that target area manually. And by doing so, then running fiber filtering, we did that in a first training cohort of the first 28 patients, randomized subset of the cohort. We found that the fornix came out, and the PAPE circuit, also the stria terminalis, which even seemed to correlate more strongly, but it kind of held true. And then this is a circular analysis now, so very circular, just to show this would this data could explain the results. But since it's circular, we built a track that explains it. That's not so interesting. When we permuted the data, it would also be better than expected by chance. But then more interesting, I think, was to leave out patients from these 28. We left out one, for example, this one here has the data point here, and that one modulated a lot of the tracks that were just defined by the other points now. And then here, in this case, the blue one, it modulated less tracks of the ones, again, not defined by itself, but by the others. And that held significant. And then we could, of course, also take the test cases. This time, we take the training cohort, define the tracts, and in this case, it modulated a lot of the tracks, here less. And again, that was a significant relationship. So it seemed robust enough for us that there might really be a relationship between where we stimulated which fibers and then also the improvements. And it did map to the PAPE circuit in general, so to begin with. And then we thought, okay, there's this other technique that that you all know, resting state fMRI. So what about polysynaptic networks? Because the one downside of diffusion tractography is it's mainly direct connections. We don't learn about the broader network architecture of the brain. So we ran essentially the same analysis. Here you just see the result. This is now what connectivity that correlates with symptom improvement. So very similar in training and test cohort, and then also in the combined cohort when we all put all together. And then we subjected this to the decoder of neurosynth that you may know from Talia Coney and Bas Poldrack, and tried to just objectively from all the neurosynth terms that are stored there to see which are the ones that are the most similar. And many of the things are like prefrontal or cingulate or so, but the functional terms that are meaningful, like retrieval was first, then memory retrieval, then episodic. Then there was one outlier, which is pain, very close by here in the cingulate, and then task demands and working memory. So it seemed like this map we found that correlates with symptom improvement when modulated was actually quite objectively associated with memory to some degree. And so this is kind of a summary figure where in the end, after doing all these cross validations, we did pool the entire data set together. And for example, going back to the tracts, this is the correlations in circular fashion of how strong the tract correlates with the improvements. But if you do leave one out correlation, it's significant with a similar correlation value, 10-fold cross-validation, significant, 7-fold cross-validation, and 5-fold and 3-fold. And that is each time really, there's no leakage there, right? So we each time have to build a new tract model, predict the left out patients, improvements. And so I've rarely seen such a robust result in the field of DBS. So we were quite excited that this actually seemed to be true. But you may now wonder, okay, but it all started with these weird flashbacks. So what's that about? Is that now also linked to this network? Because in a way, we don't want our patients to always have flashbacks, right? So in a way, to me, flashbacks are different than memory improvement. And so we could also run the stimulation sites that led to flashbacks and contrast them with the ones that did not. And there we had much more data because they tested a lot of different sites in the operation room. And drum roll, out came not the fornix, but a very different network, which is the anterior commissure of the brain. So you may know this as something that's loosely the origin of the MNI space, the 000 point, or at least in the functional domain, it is really the origin. But what is the AC really? And we didn't know either. And looked into anatomy books. I was surprised. I didn't even know this, that it would connect the temporal cortices because it's a midline structure, but it seems to really map to the frontal poles and temporal poles and temporal cortices. And then when we thought temporal cortex and flashbacks, it rang a bell. We had to think hard what the bell exactly, but at some point came to, yes, while the pen field stimulated there electrically and then induced flashbacks back in the day. So from Penfield's team stimulated almost the entire cortex of the brain, right? Across, I think, decades of work at the MNI. And these sites here, from all of these, sites, beautiful review by Penfield and Perrault, so it's definitely worth a read. But from all the simulation sites, these were the ones that led to flashbacks. And again, they were everywhere. And if we, draw in a tract there and compare that with this result we found, it's remarkably similar. So anecdotal comparison, but still, it seems like the flashback effects they observed in the operation room were a very different thing than what in the end actually happened, and they might not even have had to do with the fornix at all. But Dr. Lozano was still right that the fornix was apparently a good treatment target for Alzheimer's disease. And so to me, this is a very nice serendipitous cascade of events and convoluted story that, often discoveries are made exactly that way. So we often think one thing, and in the end, we're even right in a way, but maybe the way we got there was maybe not straightforward. So to me, a very remarkable story to have been part of. Now, we also think, can we use this technology now to, the story I just showed you was maybe more about treatment target, right? Where do we have to place the lead? But we can also think, can we use this for neuroscience? Can we use it to dissect the brain and to study the brain? And so what struck me when I entered the field of deep brain simulation around 10 years ago, that there's a small structure in the brain called the subthalamic nucleus. It's a centimeter long, small one. And if we put electrodes in it, it can help patients with Parkinson's disease, dystonia, Tourette syndrome, and even OCD. So 4 very different disorders. And you'll probably now think, oh, then this is all, all BS or not true, because it can't be that we stimulate the same region and it helps so many different things. That can't be right? Maybe it's all placebo. And it really struck me, why is that? And it's the key target for Parkinson's. So there's no doubt, right? We can switch it on and off. We really see the tremor stops. So it's not placebo, 100% not. High level evidence, New England Journal papers about it, improves quality of life and so on. And then I think we have also quite good evidence in OCD, which is very different. And we already knew before that in Parkinson's, we are a bit more in the posterior part of the nucleus, and in OCD, we are more in the anterior part of the nucleus. But it was still weird to me that the same structure could help these different disorders. So we set out into a bigger collaboration with lots of people, because guess what? There's no single center that treats all of these disorders with the treatment, but each kind of have their favorite, and many do Parkinson's, but not so many do OCD, not so many, even fewer do Tourette's, and even fewer do dystonia. So we had to essentially pool from very different sides the data sets together to analyze them together. And this is, I think, major work by Barbara Holunda and then also Ningfei Li to localize all these electrodes, put them together, analyze the data set, and so on. So the first thing we learned is that if we just look at local anatomy, where's the optimal site to stimulate, this seems to follow a gradient. So for dystonia, we have to be in the back of the nucleus, and for OCD in the front. And it seems to clutter nicely. You even somehow think, what's here in the middle, right? What's in the gaps? So which disorder would that be? But it really seems like a gradient. And if we just look at the tracts that are connected to these electrodes, we don't see a clear difference. They seem to really connect to the frontal cortex. So this is where the frontal cortex projects broadly to this nucleus. But if we apply fiber filtering, a gradient seems to emerge that looks a bit like this. So different parts of this, frontal projection to the nucleus seems to most respond to improvements in various disorders. So we could then also look at which are the cortical sites that project to that, and then that would lead to the sweet spots in the subthalamic nucleus. So in a way, this could be used to dissect the frontal cortex and learn more about it, but then also clinically to know where to put the electrodes, but then potentially also going towards a concept that we began to call a human dysfunctome, with the idea that, that I introduced in the beginning, that if we think of these circuits being the dysfunctional ones, or likely being the dysfunctional ones, then we have to lesion or downregulate for symptoms to improve. Then in a way, this starts to become a definition of potential pathways that can become dysfunctional in the human brain, and then would lead to specific symptoms. Now, how exactly they become dysfunctional, we don't know, right? It could be they're overactive, they're not transmitting the information correctly, whatever. But it seems like they're faulty, and lesioning them or downregulating them helps. So we thought this might, in a way, this is a new concept where we could try as a community to store and identify these circuits and put them into a certain library that could then be used to help specific patients. So I'll focus a bit in the remaining of the time on two of the disorders. One is OCD, and in OCD, this frontal tract that I've shown you has been a dear friend for the lab for a long time already, because we had a first study that would look at patients only with OCD with two different targets. One is in the STN that we just talked about. One was in the more frontal cortex in the anterior limb of the internal capsule. And again, we looked at connectivity between the two, seeding from the two. There you get a different profile. It's not surprising. Different parts in the brain are wired up differently. But if we use fiber filtering, the same tract emerged independently in both of them. So it seemed like, here it was more superior to the electrodes. Here was right in the STN, but it's the same structure. And in fact, we could, calculate the tract based on the ALIC patients and then estimate the outcomes in the STN patients. And that was significant with, as a cross prediction between targets. And the other way around, we could train the tract or calculate the tract just based on the STN regions, use the stimulation fields of the other cohort, and then, estimate their improvements. And that seemed to correlate significantly with the empirical improvement too. So I think at that time point, that was the first time where we cross-estimated across DBS targets using the Connectome. And then, we were excited about it, showed it at conferences. The field kind of became excited about it too. Collaborators added more data. We were able to, replicate this in 2 additional cohorts. Super excited when the New York team independently replicated this just a few months later. And then in total, there were up till now 4 replications by other groups that essentially showed the same thing. So it seemed to really hold true. The last thing we did was in a collaboration with Samir Sheth and the University of Texas. And Samir sent us 10 additional DBS patients worth of neuroimaging data, but we just had neuroimaging. We just knew the images and we knew how the electrodes were stimulated, but nothing else of the patient. So we sent it through the tract model and then could predict the clinical outcomes and sent them back to Texas. And that was a very nervous email to send because you can only do this once, right? Either you're right or wrong. You can't, check again or tweak the parameters or whatever. So sure enough, Samir's team knew the actual clinical outcomes and could compare our predictions with the real ones. And both teams were super happy that indeed it did correlate. And then another email was fantastic where a team in Brazil by Paulo Francischini and Eduardo Alio had essentially implanted in a severely affected patient with OCD electrodes that would directly modulate this tract. They switched it on, and even after just 30 days, there was a marked improvement in the symptoms that we rarely hear of, 77%, and especially not in that time frame. And the patients continued to do really well. So, but I wanted to zoom in. This was more, believe me, this tract works, but now I try to zoom in because every patient is different. Some have more obsessions, some have more compulsions, some have more depression, anxiety. So we had have another, it's an ongoing collaboration, still ongoing with lots of patients that again, Barbara Holunda curated together, lots of electrodes. And essentially these are the active sites. Essentially she wants to break up this circuit now into different domains that are more associated with improvements in obsessions, compulsions, anxiety, depression, and global functioning to, essentially further investigate the dysfunctome by going into specific symptoms that live along that gradient somewhere. In Parkinson's disease, we can do similar things. And Nanditha Rajamani has just got her paper accepted in press that looked into that. And here we could further segregate the Parkinsonian loop into specific domains that code maximally for tremor, bradykinesia, rigidity, or axial symptoms improvements. So you're beginning to see the picture where we aim to get better and better at segregating the dysfunctome or the connectome into dysfunctional circuits. And yeah, there are specific details here for the enthusiasts that make a lot of sense. But yeah, more from a clinical point of view, we could maybe think of it that way, that this could be deep brain stimulation right now, where we have an electrode with one stimulation field switched on that tries to treat all of the symptoms. But maybe in the future, we better understand exactly what we want to stimulate, and we can, on these newer electrodes, even activate two sites for two different symptoms that are most burdensome for the patient. And to help with that, we have come up with an algorithm that we call ClearTune that would test all of these different settings in silico, and can then find the optimal setting for a given patient, depending on their symptoms. And as you can see, if you run this in different ways by weighting different symptoms more strongly, you would get slightly different answers. So for example, in tremor, you have a lot of the green track because that codes for tremor. But if you weight axial symptoms more strongly, you would get more of the yellow track, and so on. So that could be the idea of using this to optimize and personalize DBS treatment. And so I hope I could, at this point, maybe convince you that trying to map this dysfunctome, or however we want to call it, might make sense, and to maybe collaborate on that across the globe to find the circuits that get faulty and that we can then dysregulate, tune down. And I want to conclude with one last study. So I know I only have 2 minutes left, but I think it's fine. Where the idea is, could we also stimulate from the outside? And I've talked about resting state fMRI before. So in the very first study, this is dated by now, we have done what I showed you in Alzheimer's, and what I showed you with tractography before. We've looked at the connectivity profile of electrodes in Parkinson's, also with functional connectivity. For each patient, we get a fingerprint. That's how it's connected. And then we correlate across these patients depending on the improvements. We had 2 cohorts at the time. The first was from Berlin, so we found the optimal functional connectivity profile, and then we could use it to estimate the ones from Würzburg, the unseen ones. And essentially this map came out, and in the second paper we could also replicate the same map for a different DBS target that also works quite well in Parkinson's, which is the pallidum. So essentially, it really looks like this is the map we need to modulate to improve Parkinson's. We know this from 2 different DBS targets by now. And if you see it like that, you really think it would be great to do it from the outside, because then we don't have to put electrodes in there. And in fact, we can, as many of you will know, there's multifocal forms of TDCS where we could fit an array of electrodes that precisely matches this network and gives you an electric field that matches the inverse of that exact network. And we did that in a blinded randomized trial where the patients either received sham or such a multifocal TDCS for 20 minutes one time, randomized, pre-registered, double-blinded crossover, blinded examiners and patients, but also blinded video ratings of the scores. And we were quite excited that the stimulation group got significantly better than the sham group, just by 3.5 UPDRS points. So that's not a lot, but it was a significant finding. And we stimulated only 20 minutes. And interestingly, out of that trial later, 8 of these patients also underwent DBS, and their effect to TDCS was very strongly correlated to the ones of DBS. So it could also be used as potentially be used as a non-invasive form of testing before we undergo surgery. What is interesting is we have lots of these maps by now. So we could potentially translate a lot of these into non-invasive forms of stimulation. You've seen the Alzheimer's map as well. So potentially this is a way forward to combine invasive forms and non-invasive forms of neuromodulation. So I want to conclude here. I hope I could convince you that using these DBS network mappings can be interesting to study also even the organization of the brain. We can map symptoms across diseases. We can map improvements across targets. And within our center here in Boston, we aim to really clinically translate these things and bring it into patients as fast as possible. If this stuff is interesting to you, I have a podcast. It's all non-commercial, so that's why I don't, I'm not ashamed to make some advertisement for it, where I interview people in the field. My lab also runs a talk series with the same name. It's all under stimulatingbrains.org. And then there's a textbook on exactly these methods that we co-authored with others. So with that, I want to thank you again for inviting me. This is my lab. I think I had a lot of collaborator slides on there too, with it's all big teamwork. So yeah, thanks to you for your attendance, but also thanks to the lab and collaborators for their help.
Thank you very much. It was really, really interesting. Let me read a few of we have a few questions, and I'll just read them to you. And if folks want to clarify, they can feel free to hop on as well. But Brad Baker asks, did you also evaluate the effect of stimulation location along the tract when performing fiber filtering?
Great question. So we did not. We could see here, for example, we could conclude from this, maybe it doesn't matter where we are along the track. It could even stimulate here or here or here, but I have really no clear information on that. I think the tract is most precise, closer to the electrodes. So especially with tractography fanning out, I would not 100% trust this part of it. Could even be that some of these loop into the cingulate, which is our hypothesis, but we don't see it because the angular resolution is not good enough. But it's certainly a testable hypothesis.
Yeah.
And it could also be that it needs to be at the place where it fans together most, but that's just speculation. I don't know.
Lots of things to try, yeah. Ardalan asks, could the disconnection symptom mapping typically used in stroke studies help identify the right fibers to stimulate across patients if high-frequency DBS acts as a lesion?
Absolutely. So I think the person is referring to Michelle's work on using the BCBT toolbox, I think, and that is very similar to what we do. We don't do it voxel-wise. We have really the streamlines in 3D, which has a slight advantage because we can hit it at different sites and have the same statistics run along the tract. But the general idea is very similar. So, Mike Fox's work on lesion network mapping is also very close to this. We co-developed DBS network mapping together. So the idea is always the same. I would still say conceptually, a lesion typically occurs and leads to a symptom, while DBS typically helps with a symptom. Although we can also study side effects, and then it's very similar. We have to rather think of this. There was maybe a fault first, and then we broke what's broken in DBS, but maybe in a stroke, it's more we break the healthy brain. So there, this connection makes more sense. Here, dysfunction might make more sense.
This is maybe related. Cyrus asked a couple questions about ultrasound. Can you replace the electrodes with a real lesion created via ultrasound? Could you stimulate with ultrasound to find to sort of test for a location before creating a permanent lesion instead of having an electrode? Yeah. I don't know. What are your thoughts on those? Yeah.
Great question. So first question was, can we do an actual lesion? And the answer is yes, right? And historically, even before DBS, people have done lesions. So this is a new thing now, actually came with ultrasound that we could call subthalamic nucleotomy. It's being done in Spain, for example, but also in some other places. Has a very similar effect to DBS. Then pallidotomies have been a clear therapeutic thing to do for decades. Has a very similar effect to pallidal DBS. It's even the same exact location. Capsulotomies have been since the 50s used for OCD and depression with variable, success, but even DBS is the same in the DBS. So we have lots of examples where an actual lesion really has the same effect. And with the same, again, there are additional effects that DBS might have that sometimes come with plasticity and so on that the lesions might not have. But the first approximation is very similar. Second question, I think, was low intensity focused ultrasound, so neuromodulatory focused ultrasound, or in the Insight Tech machine, we can also test stimulate before we lesion, and people do that. So we have an ultrasound machine here where we burn lesions, twice a week, I think, for tremor. And typically there is a test sonication first. We see the tremor stop before doing, like, we heat up and with we, it's the surgeon. So I'm sometimes, involved, but it's not me doing this. Reese Cosgrove is the key person doing this here. They heat up the tissue a little bit. Tremor often already gets better, so we know, or they know they're in the right spot, and then they burn a small hole there. And then with maybe the more research type single transducer FUS machines, I think there's a lot of promise there, but that's still very early days, right? So, but that could really be fantastic opportunity to go deep into the brain, modulate the same circuits, test before doing DBS, or even, fix some problems. So I think there's a lot of potential. I'm not doing that myself, but I think it's fantastic to try that.
I had a question about, I guess, two things. One is, what is the impact of potential confounds on the virtual lesion type analyses? And you may have done this, did you look at, I know you've showed the maps are different side by side. Did you sort of quantify how different they are? Maybe there's some unique aspects that you could isolate for one versus another location or scenario. Just curious about that. Like it's like the essentially like the partial correlation question there. Or
great question. So when we use fMRI and we for each of the electrode pairs for each patient, we run a fingerprint. We do this across 50 patients. Then I have not stressed this enough, and I should have. We often use normative connectomes even for that, because often patients don't have their own fMRI data there. Then they often look very much the same, right? They're just slight differences between them, and we then correlate across these fingerprints with the symptom, and we find something. That's one strategy. I think what you were suggesting would be rather to, in the same connectome, use both of the VTAs or both of the stimulation sites of two patients, and then use partial correlations to see the difference controlling for one another. We have not tested that at all. I know Mike Fox has done something similar in one paper with Martin Reich, but it might not be published as that, where they had two simulation settings. One led to cognitive side effects, the other one did not, and then they used partial correlations to see the difference. That was in the same exact patient. I would not know how to do it with, let's say, 50 electrodes. Can we do, one could do the inversion matrix stuff, right? I've actually thought about that, but we have not done that.
Or like you could look at variation amongst all of the differences or something, right? I mean, I don't know.
Yeah.
There's lots of things I guess you could play with. But,
yeah, people like you could do much smarter things there. So it would be great. Yeah.
Great. And I think Cyrus had one more question about, do you see how could this work impact the use of neuroimaging in a clinical setting? I think it's obviously headed in that direction, but what are your thoughts?
Great question. So I mean, one thing, sometimes a single paper rewrite might even have direct translation into some sort of clinical sense, even though, with caution, which I find very rewarding as a researcher. So in a way, if we define a sweet spot, that often already helps potentially indirectly surgeons that read it to see, maybe we should be more careful on the left side, to go more anterior, because that seems to lead to depression, right? In such a sense, we might already have had some sort of indirect impact just because it's published and the surgeons can read it and make slight alterations to their protocol. That doesn't mean, you know, it's big differences. And I think in that sense, the barrier, even though it's an invasive technique, if we slightly refine a target instead of changing it completely, it can be done that people just know better what to do, right? So that's helpful. Now with the maybe last point I said, remember this clear tune idea. So these videos there, that's something where we actively want to go into helping, you know, trying it out prospectively, where a given patient, we have the model now, a given patient has the electrode implanted already. We can run the model, we can set, you know, the weights of how much bradykinesia versus rigidity or so the patient has. The algorithm gives us one setting, we test it, and we can contrast that with the conventional settings and test that. So that's ongoing, and you know, we hope to at some point even, you know, get this into patients. We're not founding a company or so that has to be industry in the end, but at least to do the ground and footwork to show it works is actively in development.
If I may ask like two questions. One is to clarify for myself. So for those individuals, are you collecting any structural functional data, or you are using so those Connectome, is it created from the subject itself or not?
So great question. And again, I should have stressed this much more. So we don't.
We don't. Okay.
I would say in 90% of our papers, we don't.
Mm-hmm.
There are some where we did it, but there are many reasons why not. Some are pragmatic. It's just, you know, often getting to hundreds of patients is often retrospective over the last decade, and people just didn't collect it. And then afterwards, collecting it is hard because there's electrodes in the brain. Patients might even be deceased or, you know, not around anymore and so on. So it's often clinical data from the medical records that we can use. And then surprisingly, almost no hospital creates diffusion scans or resting scans. And if they do, it's often, you know, very bad quality. Then some of these patients move, right? Movement disorders patients would move. It's very hard for them to lie still. So even if you had the resources in the prospective arm to scan them well, it's much more tricky. And then, you know, some of these tracts, that's what I alluded to in the very beginning here. Some of these more finer tracts, like here, the ansa lenticularis or so, they're even very hard to reconstruct from post-mortem scans even, right? I'm involved in some of, you know, Anastasia Yendiki's work, for example, and we're trying to reconstruct these tracts, but they're hard to, even in the research grade environment, to reconstruct, but they do matter. They really play a role. So we often have to rely on high quality atlases that could be from the HCP connectomes, but also sometimes, in this case, this is not diffusion. This is really manually drawn, right? Now, again, I don't think that's the final answer, right? We have to move into individual patients to some degree, definitely. But so far, we can still learn so much using Atlas resources. And I think that's why we always do the cross-validations in our studies, right? It has to be predictive to unseen data. If not, it's, you know, just, it could be just wrong. But that's our way of, yeah. Sorry.
Yeah, thanks for further clarifying. And one more question. So if folk are interested to doing these, is there a way that they can get a hand of these data? Should they contact you? Is it publicly available?
A great question. So I'm not at the source either. I'm typically asking clinicians myself. I'm, you know, not seeing many patients, but there are many people doing DBS in almost any big university hospital. And people are, you know, the clinicians are often happy to collaborate. We have made, you know, it's hard to do, but we have made some of these available, like the Alzheimer's patients data is openly available, and the recent Nature Neuroscience paper too. But they have been published already. But if you have, you know, fancy cool new things to do with it, they are there. And then my understanding is, you know, if you approach people, there are actually many sites that do it. They are typically open to collaborations.
Thank you.
Yeah, sure. DBS is openly available, so the software is there.
Yeah. Great. Well, thanks. Thanks so much for joining us. I think we're right at 2 p.m. So we'll go ahead and let you get back to your busy day. appreciate your time and look forward to seeing your work as it evolves. It's, I think, really, really exciting. So
Thanks a lot, Vince.
yeah. Okay.
Thank you.
All right.
Good to see you.
Have a
Bye-bye.
great weekend. Okay.
You too. Bye-bye.